A very good afternoon, honorable guests, ladies and gentlemen, speakers, and good day to our friends all over the world joining us live through the live webinar platform. A big welcome to Masterminds Masterclasses, organized by the Council of the Hong Kong Laureate Forum Limited, also known as the HKLF. My name is Marcus Kwok. What an absolute honor to be part of this wonderful event. So the HKLF aims to promote understanding and interests of young generation in science and technology, as well as to foster the exchanges among scientists of different generations, cultures, and disciplines. By organizing the Hong Kong Laureate Forum, the HKLF invites recipients of our homegrown Shaw Prize and global renowned scientists to have direct academic exchange events with selected young scientists from around the world. Before the launching of the inaugural Hong Kong Laureate Forum in November 2022, we have embarked on a series of pre-forum events to cater for a variety of audiences and to further the HKLF's mission of providing a platform for scientists with different expertise worldwide to come together to promote science and to enlighten the younger generation in the scientific community. The Masterminds Masterclasses from today till this Thursday is the second chapter of the whole prelude to the inaugural forum event series. Now, to kick off the Masterminds Masterclasses, may we now invite Professor Timothy Tong, Chairman of the HKLF, to give us an opening speech. Professor Tong, please.
Good afternoon. Indeed, it's really a pleasure to see all of you here. As you heard earlier, the inaugural forum for the Laureate Forum was supposed to take place this year, but for the obvious reason, it's been postponed to next year, the same week in November next year. So mark a calendar and look forward to it. But despite the fact that we have postponed the inaugural forum, our effort to promote science has not stopped. That's why we have this Mastermind Masterclasses this week. Indeed, we have a series of activities as a prelude leading up to the inaugural forum, as I said, scheduled for next year, this week in November. But today's Masterminds Masterclasses is offered in order to bring outstanding scientists to our young people in Hong Kong, as well as those who are attending this seminar online, and so that we can build up the interest leading to the inaugural forum. And you heard earlier that this is the second chapter of our prelude to the inaugural forum. The first chapter was the science exposition that took place in September and October this year. And we had hundreds of young people coming to see the exhibition. And today, we have invited really outstanding scientists to share their insights with you, with the aim to stimulate our young people so that they may decide to pursue science as a career in their future. And today, not only that we have present audience who are present in this auditorium, we also have hundreds of people who are online watching this webcast. And today, not only we have outstanding scientists who are present in this auditorium, we also will have Professor Boncello, who is from Sweden. He is associated with Karolinska Institute, and he would be zooming in, appearing on the screen, along with Professor Iwan Hong and also Professor Dennis Low. Both are outstanding scientists locally. And so I believe that you are in for a real feast in science. So without further ado, I want to turn the floor to Professor Lep Chi Chui, who himself is also an outstanding scientist. He is known for his work in genetics, and I'm very pleased to also share with you, perhaps you know, that at one time we were fellow presidents of 2 local universities. So with great pleasure, let me introduce Lepchi Choi to stage. Lepchi?
Thank you very much, Professor Tong. Please have a seat and continue to enjoy our program today. And here we have Professor Chui Lapchi joining us in person, and Professor Chui is going to be our moderator for the panel discussion as well later. Ladies and gentlemen, Professor Chui.
Thank you. Thank you, Professor Tong, for the kind introduction. I mean, I echoed Professor Tong in his speech that this is really a warm-up session for even better science to come. Not saying that these are not scientists, not good scientists, also excellent scientists, but we do have more coming. So that he keep on telling you that there's a period to this inaugural ceremony. But first of all, I'd like to end my welcome to today's Masterminds and Masterclasses on life sciences and medicine. I'm sorry that I must read from my notes because I have the pleasant task of introducing today's 3 distinguished speakers, since each of them has made significant contributions in their specific field of biomedical research and that impacts our lives. Their bios are very long. Unfortunately, I have only 5 minutes to do my job, so I have to read instead of telling you jokes. So for Hong Kongers, the 2 guests on stage later on probably need no introduction as they appear frequently on TV and in various media channels. They are both trained as medical doctors and are now clinician scientists working on cutting-edge scientific research. First, Professor Dennis Low is the director of the Li Ka-shing Institute of Health Sciences and professor of clinical pathology and associate dean research of the Faculty of Medicine of the Chinese University of Hong Kong. Professor Low has won many accolades for his groundbreaking work on the discovery of cell-free fetal DNA in maternal blood and the development of non-invasive DNA-based prenatal testing, making such testing a clinical reality. The non-invasive DNA-based prenatal testing has been widely adopted in dozens of countries and used by millions of pregnant women every year. His recent major awards include the Breakthrough Prize in Life Sciences 2021 and the Royal Medal in Biological Sciences. He has also pioneered a number of diagnostic applications of plasma DNA in other fields, including the detection of cancer. Professor Low will be sharing with us today on the journey from scientific discoveries into clinical reality. No doubt many of the participating youngsters or young scientists would be extremely interested to find out how to make their research impactful to mankind. Now, the second panelist speaker, Professor Ivan Hong, is currently Ru Chen and Helen Lei endowed professor in health sciences pedagogy, professor of medicine and assistant dean admissions and chief of the division of infectious disease, Department of Medicine, Li Ka-shing Faculty of Medicine, the University of Hong Kong. For many of us in Hong Kong, Professor Hong is known for his association with COVID-19 vaccination. As a co-inventor of a Hong Kong government panel on vaccine reactions, Professor Hong has been sharing his expert opinions recently on the vaccination of children. Apart from his expertise on existing COVID-19 vaccines, he is also heavily involved in the development of an intranasal COVID-19 vaccine, currently under phase 1 clinical trial, as well as the forefront of clinical treatment of COVID-19 patients admitted to the hospital. His research focus, indeed, has a much wider scope. It is on treatment of vaccinology of respiratory virus infection, which includes COVID-19. Professor Hong will share his research work on various respiratory virus, antiviral treatment, and vaccinology, and his take on what is next regarding the pandemic with us. Now, the third speaker, the third guest, Professor Gonzalo Castello Branco, will join us from a beautiful city of Solna, Sweden, online. Professor Castello Branco is a neurobiologist at the Department of Medical Biochemistry and Biophysics at the world-renowned Karolinska Institute. Apart from being synonymous with the Nobel Prize in Physiology or Medicine, Karolinska Institute is also renowned for its research on cell therapy, cancer, inflammation diseases, regenerative and reparative medicine, to name but a few. Professor Castello Branco has made groundbreaking discoveries about how the cells in the brain works. His work is particularly important in the development of new treatment for neurodegenerative disease like multiple sclerosis, where the current treatment focuses on inhibiting the immune systems from attacking the oligodendrocytes that produce myelin to direct the nerve impulses. Now, his presentation will be on the function of oligodendrocytes in neural development and disease. Now, without further ado, may I now invite Professor Low to come on stage, please.
So I'd like to thank Professor Timothy Tong for the kind invitation. It is my great honor to be able to stand here in this distinguished forum. So today I'd like to share with you some of the stories that transformed me from a medical student to become a clinician scientist. So this is a picture of myself, actually taken over 30 years ago when I was a medical student at Oxford. Now, so as a medical student, we have to study different subjects. And one of those subjects is obstetrics. And so I learned about the process of prenatal testing, which at that time actually involved invasive technology like amniocentesis shown here, in which a long needle is inserted in the uterus and some fluid is taken. But every time you do that, there's a chance that you will actually kill the baby. And so as a young medical student, I was wondering, is it possible to actually do something safer just by taking blood sample from the mother? So I was thinking, is it possible that during pregnancy, the baby might actually release its cells into the bloodstream of the mother? But you want to do that, we have to have some ways to distinguish the fetal cells from the mother cells. So I was thinking about a situation, if the baby is a boy, then maybe it's possible we can differentiate those cells. So actually I managed to persuade a professor to let me actually spend some time in his laboratory. And then we do an experiment like this, in which we have a dye which will actually stain a male chromosome. And every time we do that, you see a black dot inside the cell. So in this slide here, you can see there are 4 male cells in the blood of this pregnant mother. So showing that indeed my concept is correct. And then also at that time, a technology called the PCR, polymerase chain reaction, which is just coming into practice. So I also managed to actually use the PCR to amplify this Y chromosome DNA. So eventually I was able to publish this paper in Lancet just a year when I qualified as a doctor. And then actually a few months later, the Lancet actually published an editorial on this called Is It a Boy? Because using this technology, you can theoretically tell the sex of the baby. And then I graduated and then became a doctor. So I worked 1 year as a houseman. But during that year, I was constantly thinking about this field because I thought that since I've started it, I have some responsibility to finish it. So after 1 year, I decided not to proceed with medical training for a few years, but to actually do it full-time to pursue a PhD in Oxford. So it's a picture of myself as a PhD student. So you can see that I'm sort of covered up like a terrorist. And the reason is because I'm actually using a Y chromosome as a marker. And of course, every cell in my body has a Y chromosome. So I'm trying to protect my samples from myself. But the problem is that once I start working on this full time, I realized the project is not as simple as I'd have thought. Because even though the fetal cells are there in the mother's blood, the concentration is very low. It's like trying to find the needle in a haystack. So after almost 4 years, I still could not actually get a system to work robustly. So eventually my scholarship ran out, so I have to graduate, so which I got my PhD in summer of '94. So at that time, the science didn't go well, but at least I met my wife, Alice, and so we managed to actually graduate together. And then I went back into clinical practice and for the next few years. And then in '97, so my wife and I decided to come back to Hong Kong. And then I joined the Chinese University of Hong Kong. And I remember actually about 3 months before I left from Hong Kong, actually I saw in this journal, there are 2 papers which says that in cancer patients, you can actually see cell-free cancer DNA in the plasma of those patients. And then suddenly I have a strange thought, because I thought a baby living inside a mother is actually a little bit similar to a cancer growing in a patient. So maybe I was also looking in the wrong place, because for the last 8 years, I was trying to find the fetal cells. Maybe the thing I should be looking at is in between the cells, in the fluid, where the cell-free fetal DNA. But the problem is that at that time, I was just about to leave UK to Hong Kong, so I don't have a grant. So, and I've never worked with plasma before. So I was wondering, how can I process this plasma? And then I remember when I was in the UK, I don't quite get used to the college food. So every now and then, I actually went home to cook some instant noodles, right? And so what I did is that you boil some water and then put the noodles in. And a few minutes later, you got your meal. So I was thinking, how about if I just boil some of that plasma and let's take a few drops of the boiled soup to be tested. But surprisingly, even in that very crude preparation, I was able to see this Y chromosome signal in some women, and whereas in other women, I don't see the signal. So eventually, we find that all the women with that white line all have baby boys, whereas those without signal all have baby girls. So this is the first discovery of cell-free fetal DNA in maternal plasma. It hasn't been shown before because previously people just look at the cell and throw away the plasma. And then we realized this cell-free DNA is present very early on in the first trimester. And actually by the 10 weeks of pregnancy, it reaches some 15%, which is amazing because imagine the baby at that time was very small compared with the size of the mother. And then another very surprising thing is that after the baby is born, within 2 hours, this DNA is gone. So as if the mother is trying to protect herself from this fetal DNA. And then we decided to actually try to use this discovery for some prenatal testing. And usually we start with simple things first. So the first thing is that we show that we can accurately determine the sex of a baby, which is useful because some genetic diseases like hemophilia typically affect boys. And then a second example we show is that we can use this to detect the blood group type of the baby. Because sometimes if the mother and baby have different blood group types, the mother could attack the baby immunologically. And so these 2 applications were then rapidly introduced into clinical practice all over the world. And then we become more ambitious. So the number 1 reason why pregnant women go for prenatal testing is because they're worried the baby might be suffering from chromosome disorders like Down syndrome. So Down syndrome is caused by the fact that the baby chromosome 21, shown in the bottom here, instead of having 2 copies, they have 3 copies. And so typically you diagnose Down syndrome by looking at a cell and then counting how many chromosome 21 you have inside the cell. And then, of course, we wonder, well, can we use the cell-free DNA approach for this? But difficulty is that in cell-free DNA technology, we don't have cells. So the DNA is just swimming outside cells and mixed in with the mother DNA. So how can you just know how many copies of chromosome 21 a baby has got? And furthermore, when you analyze the whole thing together, you can look at here, the red dots are the mother's DNA, and the yellow dot is the fetal DNA. So the mother DNA is going to overwhelm the fetal signal if you just analyze it together. So I actually spent 10 years trying to solve this problem. And eventually, I realized that the way to do that is instead of analyzing everything together in one test tube, is to separate the molecules into many, many test tubes. so that one test tube only have one molecule or no molecule. So you transform this into a digital format, like in computer, the 1 and the 0. So in this sort of situation, the fetal DNA will be present on itself without interference of mother's DNA. And so we cracked this in 2007 and published this paper. But surprisingly, within a few weeks of our publication, there's another paper from another group on exactly the same thing. And also very strange is that two papers were submitted on the same day, so May 9, 2007. So you can see that it's very strange. Sometimes in science, if you think of something, somebody else might also think about that. And this competition is what makes science very exciting. But anyway, so this digital approach has advantage, but it also is not perfect. And one reason is because we focus on one gene on chromosome 21, and there are many genes on that chromosome. So we're losing the information of those other genes. So in this sort of counting, the more counts you've got, the higher the statistical power. So I was thinking, the way to improve the accuracy is to count millions of those DNA molecules, which we can do by DNA sequencing. And because we know the human genome sequence, I can map each of those sequences back to the chromosome from which it comes from. And in the bottom here, you can see I can count the relative ratio of different chromosomes. And interestingly, by doing that, we're able to actually detect the chromosome 21 signal here on the left in Down syndrome babies was very high. The signal in green and red, much higher than those in normal baby, which is in blue and orange. And actually, indeed, in an initial series of 14 Down syndrome pregnancy versus 14 controls, we have 100% accuracy. So very excitingly, we published this paper in 2008. But once again, within few weeks of this paper, there's another paper from the same group. And so I wonder, so who is this guy who is constantly competing with me? So I look him up on the website, and I find that he's a person called Stephen Quick, and he actually works in Stanford. But an interesting thing about him, he actually studied in Oxford, and also actually graduated in the same year that I did, which is 94. So then I went home and told my wife about this. And interesting, she actually went back and took a look at this picture. So you can see myself there, and Steve Quick is over here. So you see, it's almost like, fate has brought us together. And then a few years later, I was very fortunate to be elected into U.S. National Academy of Science. And on the day when the result came out, I saw my name, so low somewhere there. And a few names down, I saw his name, Quick. So this was actually taken at the admission ceremony when the three of us, remember what we stood during the graduation ceremony. But anyway, so back to the science. So now we have this prototype method, which seems to be very accurate to detect Down syndrome. So then we spent the next 3 years doing a clinical trial to see how good it actually is in practice. And interesting, actually, we find that the accuracy is 99.7%. So we published this in 2011, and we feel now the technology is actually ready to be launched into clinical practice. But the problem is that I couldn't do it in a university, right? I have to work with some companies. But company won't work with you unless you actually have a way to protect your technology, like through patents. So for the last 20 years, we have also learned about patenting. But initially, we weren't very good at it. So for example, one of the first patents that we have filed, our university licensed it to a company based in the UK. But they didn't realize the importance of that patent. So they sat on it for 3 years and just gave it back to the university. They thought it was rubbish. But of course, eventually, that patent was actually very valuable. And then I remember, then after that disappointment, I then went on a conference to Thailand, and I met this gentleman who is Charles Cantor, who at that time, he was the was chief scientific officer of Sequenom. And then we start talking, and eventually, actually, Sequenom licensed our technology. But on the other side, of course, in Stanford, their professors have a lot of experience with commercialization. So Steve Quick actually licensed his technology to a company called Verinata, which was then bought by a bigger company called Illumina. So now you have two companies, Sequenom and Illumina, having the different patents, and they start to fight, as they usually do. But eventually, fortunately, they decide to settle it out of the court. And so the Stanford and my patent were pooled together, and then people can access that patent pool easily. So anyway, the technology was launched in the autumn of 2011. So it's now called NIPT, so non-invasive prenatal testing. It's now available in 90 countries around the world, and millions of cases was done every year. For example, in China, now about 1/3 of babies born undergo this testing. But how about Hong Kong? So because this is my home city, of course, I want that we have the best service possible. So part of our funding came from the Innovation and Technology Commission. So the previous commissioner, Janet Wong, once met me and said, well, why don't you go and open your own company? So together with a number of our students, then we started a company called Exelon, which stands for Extracellular Genome. And actually, the Chinese name may be more informative. It's called Nga Si Lang. Being Nga Si is like scholar, right? So even a scholar can do it. And then Exelon becomes a market leader in Hong Kong in this area. But of course, having all this thing in the private market might not be ideal because some women might not be able to afford to test commercially. So in 2019, we basically gave the patent free of charge for a high-risk woman to the hospital authority. So now they're offering this free of charge from the Children's Hospital in Kai Tak. So it seems that the goal that I started my journey has almost been done. But what else can we do? Now, so if you recall, one reason why I discovered fetal DNA in maternal plasma is because I was inspired by the work in cancer. So now that the fetal side is going so well, maybe it's time to actually try to see if our technology can benefit cancer. So one cancer we start off with is this cancer called nasopharyngeal cancer, which is a cancer in head and neck. And this cancer is very common in the southern part of China. For example, a Cantonese man like myself have a lifetime risk of this cancer about 1 in 39. Another reason why we work on nasopharyngeal cancer is because the cancer is associated with a virus infection, Epstein-Barr virus. So about 20 years ago, I was thinking, well, maybe in the course of its biology, this cancer cell will release the EBV, this virus DNA, in the bloodstream of the patient. And I can use this virus DNA like a marker, just like the Y chromosome from a male baby. And when we did experiment, we find that on the left here, the blood of this cancer patient really stuffed with this virus DNA. But on the right-hand side, in healthy individual, most of them are negative. Then I wonder, well, can I use this to detect cancer early? Now, I think it's worth doing because if you look at this survival figure, so every time somebody dies, the curve will dip down. So when you look at the top graph, which is if you discover cancer in stage 1, then when you're long term, over 90% of the patients will survive. But you go to the bottom graph in stage 4, then maybe some 40% of them will die. But unfortunately, in Hong Kong, about 76% of this cancer discovered in stage 3 and stage 4, and many people were dying. So we think that we'd like to see if I can use this blood test to detect the cancer earlier so they can do something about that. So for a period of 3 years, we decided to look at maybe some 20,000 Hong Kong citizens. We'd like to focus on the highest risk group, which is males between the age of 40 to 60, and see if early detection is possible. So every week, we send a team to different districts in Hong Kong. So eventually, we reach our goal, and we find that there are 34 cases of this cancer. And this is interesting, because if you calculate from the incidence of this cancer, in 20,000 people, there should only be 7 cases of cancer, and yet we have 34. That means that we're basically detecting cancer, which is going to present itself for the next 4 to 5 years. And indeed, because of that, we should have an early detection. Now, for example, without screening, you can see here, most of the cancer is discovered in stage 3 and stage 4. But now, with this new technology, we change it so that most of the cases is detected in stage 1 and stage 2, so the green and blue. And also, when you follow those individuals up, you can see that in the top graph, for the 34 cases, only 1 died. The rest basically is cure of disease. In the bottom, if you don't do the screening, then the chance of them dying is 10 times higher. So this work was published in 2017 and was chosen to be one of the most notable papers of New England Journal in that year. And actually, after a few years of work, we now actually have created a product based on this test, which hopefully can eventually save life in this area. But nasopharyngeal cancer is only the first step. So we'd like to actually generalize that technology to other cancer types. And to do this, we need to develop a type of marker, cancer marker, which can be applied to every type of cancer, not like the virus DNA, which can only be used in a subset. So eventually, we actually develop a test which is based on epigenetics. So epigenetics is like modification of the DNA, okay, like the decoration in this room, like the furniture. And we find that by using this epigenetic test, we can basically detect multiple cancer in one go. And furthermore, not only can we detect the cancer, because different organs in our body have different epigenetic signature, I can also use it to tell us where is the cancer likely to be. So now with those two things, we believe that we have a test which can save lives. So from the story of NIPT, then we know the best way to do that is to have our own company. So together with two of my colleagues, Rosa Chiu and Alan Chan, we started a company called Serena, which stands for circulating nucleic acid. Basically, we're trying to develop products who can detect cancer in the early stage so that we can do something about that. But surprisingly, in the Silicon Valley, somebody else have also started another company called Grail. And you can see the mission is very similar, is to detect cancer early when it can be cured. So basically, you can see that Serena and Grail are moving in the same direction. So from my previous experience, if that happened, then lawsuit is what you'll get, right? So this time we're wiser. So in 2017, we decided to talk with each other. And then the two companies actually merged together, and I've become a scientific co-founder of Grail. And this is actually in a party to celebrate the merger. So the guy next to me is the first CEO of Grail. He's a person called Jeff Huber. He's the one who actually brings you Google Maps. And so eventually, after a few years of work, we now have a product which can test for 50 different types of cancer together. So the FDA regards this as something called a multi-cancer early detection test. And hopefully, this will be a weapon against this biggest killer in the world. So finally, as a summary, I think that if you are interested to do research, it's worthwhile to think about something which is not obvious. Like when everybody's looking at DNA inside cell, I decide to look at DNA outside cell. And also, it also helps if you can see some connection between apparently different subjects. Like you can see a connection between pregnancy and cancer. And also, it's useful to have people who knows both medicine and science, the clinician scientists. Because the doctor side of me will tell me what the patients need, but scientist side of me will tell me what is actually possible in the laboratory. And finally, there's a lot of synergy between commercialization and academia. Ultimately, we can create an ecosystem which can push everything faster. Also, I'm very fortunate to have a very nice lab team which have helped me and worked with me for the last 20 years. So thank you very much for your time.
Thank you, Professor Low. Professor Low, please remain on stage as we're going to have Q&A question now. Very interesting presentation. A lot of good news, early cancer detection, and a lot for us to take in as well. Now, shall we start the Q&A by taking a question online, if that's okay?
Sure.
All right, so first question online. How close are we to screening cancer on a large scale by cell-free DNA or RNA analysis? Professor Low.
So for example, like I mentioned just now, that the test from Grail actually was launched in the US a few months ago. And they are also doing a trial in the National Health Service in the UK. And also, if you look across the world, there are many other companies in this space. For example, a company called Thrive, which come from technology from Johns Hopkins, and many other companies based in China. So I think we're very close. I think what is lacking now is to show that for this multiple type of cancer, the screening will actually reduce mortality, just like what we have shown for nasopharyngeal cancer. So we need to show it for multiple other types of cancer.
Right. Still, good news.
Yes.
All right, now a question from the floor. Hong Kong here. Now is your chance. Please take this opportunity. Simply raise your hand and our lovely staff will pass you the microphone. We got a question here already. A lady wearing a black jacket at the front, at the second row. Hello.
Thank you for sharing, Professor. So I'm just curious, for this multi-cancer screening project that you've been working on, what's the false negative rate on it? And what do you think can be done to continue to improve the product?
So currently, if you look at stage 1, stage 2, stage 3, stage 4, so in stage 1 overall is about 25% sensitivity, and stage 2 is about 40%, and stage 3 and stage 4 then will go to 70 or 80%. So clearly there is room for improvement in the stage 1 and stage 2. But my thinking is that even detecting 25%, you are already helping that 25% of patients who would normally don't know they have cancer. They will already seek earlier treatment. But of course, the question now is how do we push that 25 to maybe 90%? So that will require a third generation of work. Now, for example, like, is it possible to have sequencing methods which are more sensitive? For example, apart from epigenetics, can we detect other markers? For example, there was mentioned RNA markers and other things. Thank you.
Okay, very interesting question. Thank you very much. Now, because we're running out of time, we're going to take one more question from online. Okay, what further new applications do you think plasma DNA analysis have?
Okay, so just now I mentioned about this epigenetic type of test, and every organs in our body will have a different epigenetic marker. So I think that, for example, with the aging population, disease of the brain like Alzheimer's and others are getting increasingly important. So one of the areas I want to move into is to use it to detect neurological disorders. Yeah, thank you.
All right, Professor Low, ladies and gentlemen. Very interesting presentation. A lot for us to think about. And next, for our second presentation, we have Professor Ivan Hung. So let's invite Professor Ivan Hung, Clinical Professor of Medicine, Assistant Dean and Chief of the Division of Infectious Diseases of Li Ka-shing Faculty of Medicine of the University of Hong Kong. Professor Hung, the time is yours.
Thank you. So thank you, Professor Timothy Tong, for very kind invitation, and also for Professor Lap Chi Choi for the very kind introduction. It's always a privilege of mine to share the same stage with Dennis. So my talk today will be on from SARS 2003 to swine flu H1N1 2009, and then to COVID-19. What have we learned so far? So let's turn back the clock to 18 years ago, when I was still a trainee. And I have the privilege of working under two very prominent scientists, Professor Monique Pierres and my mentor, Professor K.Y. Yun. So you can see that basically Professor K.Y. Yun and Professor Monique Pierres haven't changed much, apart from, in fact, they got more hair than before. So at that time, it actually came as a surprise that there's an outbreak of the SARS 2003 from the Guangdong province. And of course, that comes in fact in early Jan and then February, where there's a cluster of pneumonia cases of unknown origin. And of course, at that time, it's a big impact to Hong Kong and also to mainland China, where there has been a lot of mortalities and morbidities. And the mortalities actually was 10% at that time, and a great disruption to the economy. And of course, at that time, I was a trainee at infectious disease, just starting my training under Professor Yun. And I still remember the day when I was actually volunteer to join the so-called the front line at fighting for SARS. And then I was in the, at that time there was no so-called isolation ward. It's a general medical ward that we worked in, and then we basically changed into the gown, the protective gown, and then of course just with an N95, without any face shield or anything. And it worked there for like 3 hours continuously inside. But basically at that time, everything was very limited. And of course, in the beginning, we don't know what we're treating. And then of course, we don't have much to tell apart from just monitoring the patient clinical presentation, the chest X-ray. And many of them, in fact, deteriorated. And the only treatment that we have at that time was giving steroids, which unfortunately many of them have a lot of complication. However, I still remember the day when we were called upon, when we were consulted by the Guanghua Hospital ICU, where Professor K.Y. Yun, and I was in the lab, and Professor K.Y. Yun said that he's going to Guanghua Hospital to look at this patient. And this patient, of course, is the relative of our index case, one of the nephrologists who unfortunately came down with severe pneumonia and admitted to Guangwang Hospital. Professor Yun went to the Guangwang Hospital and, of course, saw that patient, gave some advice. And most important is that from that patient, he bring back basically the lung biopsy of that patient before he was transferred to the intensive care. And from that specimen, I still remember after coming back, he discussed with me and Professor Monique Pierres and Dr. Kay Chen what could be the possibility. He actually lists out all the possibilities of causing this pneumonia, and one of them, of course, is coronaviruses. And from that, of course, Professor Monique Pierres and our team was able to isolate and identify the SARS coronavirus. And we were the first team to isolate that virus to be causing SARS. And that was published in The Lancet, of course. And subsequently, of course, within a day, this finding was confirmed by the German and also the US group. And you can see on that, on the diagram on the right, there's in the electron microscopy, the coronavirus on the alveoli of that patient in a culture. And then, of course, we also able to find the antibody towards this SARS coronavirus. After that, we had the chance, of course, in working. After finishing work, there was an outbreak in Amoy Garden, and hundreds of patients was admitted to United Christian Hospital. Myself and Vincent Zhang and also Bong Tang, the three of us were sent to United Christian Hospital after work to see this patient. And we spent 3 days working non-stop and saw every single one of these patients, examine the chest X-ray, examine them. And then, of course, with us, we were able to take back many of these nasopharyngeal aspirate, many of these stool and urine sample. And then from that, we are able to work out the actual pathogenesis and also the viral progression with respect to the clinical presentation. And we're able to define, of course, that is actually there were 3 phases in terms of the SARS. And the first phase, there's a cytolytic phase, where there's a replication of the virus, followed by an immune response in terms of controlling the virus. And then, of course, there's the convalescent phase. And then, of course, different from influenza is that the viral actually peak in the 2nd week, rather than the first few days from symptom onset. And that, of course, very different, and also give us time, in fact, to treat this patient in the first week if we're able to find something that works against the coronavirus. And then, of course, the other things that we're trying to work out is what happened in Amoy Garden? How come that so many patients actually came down with SARS within a very short period of time? And then, of course, what we did is that we were able to match the viral load. We find that basically there was a case in the Block E, Unit 8, and there was a patient who unfortunately came down with SARS, and is a patient on peritoneal dialysis. So he was actually emptying the dialysate with lots of virus into the toilet. And then he put on the fan, and of course, because of the U-shaped pipe, the virus was actually drafted back and then into the air. And because of it becomes an airborne situation where the virus was actually spread to various units in the Amoy Garden. And then what we did is that we matched the viral load of these patients and where they're actually living with respect to the index case. And what we find is that basically patients who live closer to that unit have a high chance of coming down with COVID, with SARS, and of course, with a higher viral load upon admission. The closer it is, and in the direction of the wind, they have a higher viral load. And then, of course, these patients have a higher chance of complication. And that came in the 2nd paper that we published in my paper published in the Emerging Infectious Disease, is that we're able to match the viral load in various specimens, and then, of course, seeing that the high number of viruses and the larger number of sample in different organs in terms of, and also clinical presentation, will result in a much more severe disease in these patients. And then, of course, subsequently, the epidemic died after a few months, but the research didn't stop there. In fact, Professor K.Y. Yun's team then subsequently decided to look for the origin of SARS. And of course, the likelihood is that we're thinking because of bats, they actually carry a lot of different kind of viruses, the Ebola, the Nipah virus, they all come from bats. So the likelihood is that it's possible that coronavirus actually come from bats. So we work with the Agriculture and Fishery Department and able to use the mist nets, you can see there, to catch the bats at night. And you can see Professor K.Y. Yun there helping to catch the bats, which he's not very good at. And then, of course, from the horseshoe bats, we were able to isolate the virus from the oropharynx and also the rectal swab from this horseshoe bats. They are very commonly found in southern part of China. And from that, we're able to find the best SARS coronavirus-related coronavirus, what we name it HKU3. And in fact, you can see that basically Professor Yu and our team group were able to identify 30 different kind of novel coronavirus over the years, including, of course, the Chinese horseshoe bat, and also that the HKU4 and HKU5. In fact, Professor Yu named it all after HKU because he loved HKU so much. So HKU4 and HKU5 is actually the MERS coronavirus, matching very closely to that. And then, of course, lately is the SARS-CoV-2 virus that was also isolated from the Shenzhen group as well. The other coincidence, of course, has been said by Professor Dennis Low, is that at the same time when we published the paper in PNAS, about a week later, a group from Singapore, Professor Wang Yingfa, also published the similar studies on identifying the best coronavirus as one of the possible origin for SARS. Then come the problem, because after SARS actually died pretty quickly because it's not a very smart virus, because it's too lethal, have a mortality of 10%. So basically what happens is that we then run out of research grant. So we have to think of something else to do. And of course, then the other things that we are interested in is doing influenza. And then, of course, the pandemic came in 2009 with the swine flu. And here is the influenza A virus that on the surface there's a hemagglutinin and also the neuraminidase enzyme. And of course, during the early days of swine pandemic, is that patients, even though they are very young, they came down with severe pneumonia, as been shown in this patient who is a Filipino domestic helper, admitted to Ritchie Hospital with severe pneumonia. And despite with the ECMO support and ventilation, the patient succumbed. And of course, the biopsy showed that is a very high cytokine reaction and also basically a very high inflammation within the lung parenchyma, as been shown here. And the study from myself and Dr. Calvin To, who's my very close friend and collaborator, is that we were able to find for those patients who have severe influenza, in fact, the viral load remains very, very high after a few days. Whereas for mild disease, the viral load actually dropped pretty quickly. And as a result of the high viral load, the patient also have a very high cytokine storm of interleukin-6 and also interleukin-10, as being shown here. So basically, is the cytokine reaction, is the inflammation that is actually killing the patient. And we know that with the antiviral treatment, like Tamiflu, it doesn't work very well. And also that you have to give the antiviral very early on, perhaps within the first 48 hours from symptom onset. And that will probably make a difference. And whether it's actually reduced mortality, we do not know. So what happened is that we decided to harvest the convalescent plasma from patients who have recovered from the swine flu, especially in young patients who actually remain pretty asymptomatic, but they are able to generate very high antibody. And from that, we are using both the convalescent plasma and then subsequently fractionated to the hyperimmune IVIG with very high antibody against the swine flu. And we published this paper by treating this patient in a double-blind randomized control trial and find that basically patients who've been treated with the convalescent plasma have a much quicker recovery, which they are able to suppress the viral load and also resulting in a much lower cytokine storm, and hence there are fewer mortality as a result. This finding was subsequently proven by a much larger study by the NIH group in 2014, 2015, where they published in the Lancet Respiratory Disease that the convalescent plasma and the hyperimmune IVIG actually works very well to treatment of severe influenza. Apart from doing that, we also look for some medications that are already available that we can repurpose in treating severe influenza, because we know that it's very difficult and it's a long time before we have something through a clinical trial, phase 1, 2, 3 trial, completing to have a new antiviral. So what we're doing is we are repurposing medications that are already available, that are used to treat, for example, some antibiotics or some other anti-inflammatory, and putting it as a combination and using this as a repurposing treatment to treat severe influenza. And what we find in the lab is translated into using this repurposing medication. And of course, we find that by combining oseltamivir, clarithromycin, which is an antibiotic, and naproxen, we're able again to suppress the viral load in influenza and also resulting in a much reduced cytokine reaction and a better clinical outcome in this severe influenza group. Besides antiviral, we're also interested in how to improve the immunogenicity of patients with influenza vaccine, especially in the elderly group. So what we did is that we initially were interested in intradermal vaccination, which of course you have seen Professor K.Y. Yun doing it to himself by the intradermal vaccination. But we're actually doing it with a proper device with a micron jet. And also what we're doing, we are applying a toll-like receptor 7 agonist before we give the vaccination. And we find that basically in this study that despite there's an antigenic drift and change in the X3 and 2, we were able to give this vaccine and able to mount a significantly better antibody response by using this strategy of vaccination compared to intramuscular vaccination, resulting in much higher antibody and also a much better clinical efficacy. So now come to COVID-19. And of course, I still remember the days in early Jan 2020 when we were first notified by the Chinese CDC that there is another unknown cluster of pneumonia in Wuhan. And again, I was talking to Professor Yun, and immediately we thought that it could very well be coronaviruses because it doesn't behave like avian influenza. And then, of course, subsequently, Dr. Jasper Chan and Professor K.Y. Yun were able to identify a first cluster, family cluster of cases who actually came down from Wuhan to Shenzhen, and then find that basically many of the family members remains asymptomatic, and they could pass the virus within the family. And subsequently, we then quickly designed a posterior oral pharyngeal testing by Dr. Kelvin To and myself that we're able to use a non-invasive way rather than using a nasal pharyngeal swab. And basically, by using a saliva detection for PCR for this viral load was actually matching that, correlating it very well with the traditional way of using an endotracheal aspirate or a nasal pharyngeal swab. Besides that, we also look at a patient who returned from the Princess Diamond cruise that we were able to, from those quarantine patients, we were able to identify, in fact, cases that have been missed by the Japanese health ministry, saying that they are negative. But in fact, we are able to find 10 patients who were subsequently tested to be positive. And from this patient, we know that one symptomatic patient behind, there's actually 2 to 3 asymptomatic patients in this cohort. The other very important findings is that we compare the pathogenesis of COVID-19 to that with SARS. And we find that basically, compared to SARS, the viral load actually peaked much quicker. If you look at the diagram in severe COVID-19, we find that basically the viral load actually peaked around day 4 to day 5 from symptom onset. And even earlier for the Delta variants now, probably day 2 and day 3. And for patients who develop severe COVID, they will have a very high viral load, followed by, in the second week, a so-called hyperinflammatory phase. So what happened is that if we are able to use some medication to suppress the viral load in the first week, then it's likely that the patient will recover much quicker and that the hyperinflammatory phase response will be much lower, and fewer patients will die with fewer so-called hyperinflammatory cytokine storm. And of course, we know that we have to identify at-risk patients, especially patients who are elderly, patients who are diabetic, patients who have got cardiovascular disease, which are higher chance of dying. And if you look at the cohort in Hong Kong, most patients who died were above the age of 70, especially the group above the age of 80. So what the principle of antiviral treatment, in fact, shared by both influenza and COVID, is early treatment. So if you're able to, in the high-risk group, if you're able to give an early commandments of antiviral, and you give a combination of antiviral, which probably are just repurposing medication, it will be able to work very well, even though it's probably moderately active. And by doing so, you're able to suppress the viral load and the cytokine storm, and also reduce the shedding and complication. And of course, we have been doing this and doing in vitro screening of these repurposing medication in the early days of SARS, where we identified Kaletra, which is a protease inhibitor, together with a interferon beta-1b that works very well against MERS. And also, we've been doing this similarly for COVID-19, and we're able to look at the EC50, where it's able to, the plaque reduction assay, and then compare to the cytotoxic, the CC50 concentration, and we identify interferon beta-1b as the best, with the best selective index, together with remdesivir, which is also moderately active against the COVID-19. And very early on, before the days we have remdesivir, we're using the triple combination of interferon beta-1b, Kaletra, and ribavirin from what we know from the lab. And we use this triple combination to treat patients with COVID-19 in Hong Kong. And what we find is that basically they have a much quicker clinical recovery, together with a much quicker viral load suppression, compared to those who are just using Kaletra as a control. And by comparison, we know that interferon beta-1b is the backbone of the treatment. And this is, of course, supported by subsequent finding from other groups that in patients with severe disease, they have a much, basically a much suppressed interferon 1 to 3, and suppressing the innate immunity, and thereby allowing the virus to go haywire and replicate very quickly. And patients who have life-threatening disease, they also intrinsically develop anti-interferon antibody. And if you, by replacing the interferon, the patient will be able to recover much quicker and able to suppress the viral load. And this, of course, there are subsequent findings, including the UK group, where they give a intranasal interferon beta-1a, or in the Toronto group, where they actually give a injection interferon lambda. And both studies show that if it's early treatment, you're able to suppress the viral load and lead to a much quicker recovery. And similarly for MERS, where in the Saudi Arabian group, again, they publish interferon beta-1b and Kaletra and lead to a much better clinical outcome and fewer mortality. Apart from using interferon, again, we were working very closely with Hong Kong Red Cross, where we're harvesting convalescent plasma from recovered COVID-19 patients. And of course, this treatment has resulted in much better recovery in some of the severe cases who was unfortunately late presenters to miss the chance of early treatment. And finally, of course, I would like to share with you one of our latest study. It's a phase 1 randomized controlled trial with the vector vaccine, influenza vector vaccine, developed by Professor Chen Honglin from our group. And we have just completed our phase 1 study in 18 to 55 years old healthy individual. And what we are looking at is that this vaccine is able to confer a very robust mucosal IgE immunity. And important of this vaccine is that not only is able to mount a confer local mucosal antibody response, and thereby suppressing the transmission and the carriage of the virus in the nose, is also able to add the influenza to this vaccine. So in the future, hopefully, if we're able to go on and into phase 2 or phase 3, we will be able to offer this as a combination vaccine of both influenza and also with, for COVID-19. So with this, I would like to end my talk by sharing this, my favorite football team, Liverpool, that of course we have to work together, like what Captain Tom Moore, who unfortunately subsequently succumbed to COVID-19, but before that he was actually singing this song with the NHS healthcare workers and we have to work together in order to fight off this severe pandemics and also future pandemics. So with this, I would like to end my talk by acknowledging all my colleagues, both from Department of Health, from HKU, and from the Hospital Authority. Thank you.
Professor Hung, thank you so much. That was a great presentation. We just want to say thank you for fighting for a better Hong Kong and, of course, the world as well. Now, because of time limitation for this Q&A session, we can only answer 2 questions, one from online and one from the floor here. Shall we take the first one? All right. Is there any positive impact of COVID-19 pandemic on medical science?
Yes, indeed. I think never ever before, as in earlier this morning, I was with Professor Choi, that in one of the discussions we actually mentioned that, of course, the positive impact of COVID-19 is that actually speed up a lot of the vaccine or antiviral development. And never in our history before that we have more than 100 vaccines for COVID-19 at various stages in phase 1 to phase 3. Some of them already licensed within a 1-year period of time. And of course, apart from vaccine technology, other positive impact, including antiviral treatment, including PPE, and also, of course, the bringing the world together for collaboration. Of course, there's still a lot of gaps in between that we have to do better, but already it's a very good start.
Yep. All right. So, a question from the floor. Yes, a lady in the middle row here. That's right. Yes, perhaps you could help us pass the microphone to... Hello.
Thank you, Professor. I wanted to ask about the names of the viruses. Like, are they made up or do they have certain names or meanings to it?
Well, basically the names, of course, you have to mention what by the classification what they are. But in terms of the individual species that you're able to identify, it's actually named by the one who actually found that particular virus. So from Professor Hill and his group find so many viruses because he basically loved HKU so much, so he basically named it after HKU, 1 to 30. So you can name it whatever you like, but of course it has to be associated with that particular virus. But the species itself, you can name it whatever you like.
All right, thank you for the interesting question. And ladies and gentlemen, thank you, Professor Hung. We'll see you again at the panel discussion. All right, for our next speaker, we are very, very excited because we have Professor Gonzalo Castelo Branco from Karolinska Institute in Sweden joining us live. Now, Professor Castelo Branco is the professor of glial cell biology at the Department of Medical Biochemistry and Biophysics. So let's say hello to Professor Castelo Branco. Hello, Professor.
Hello.
How are you?
To be here. I'm very
Hi.
good.
You're live from Sweden. What's the time there now in Sweden?
Now it's 11 a.m. in the morning.
Oh, nice and early.
Yes, exactly.
Fantastic. All right. We've all been waiting for you, so the time is yours.
Okay, thank you very much. I will share my screen and put my laser pointer. So I would like to thank, first of all, the organizers for the opportunity to present our work in this symposium, and for which I'm very honored. So our lab, we work in oligodendrocytes. And oligodendrocytes is a glial cell type in the brain and in the spinal cord that produces myelin, which is this lipid-rich membrane that insulates the nerve cells and thereby allows fast transmission of electrical impulses by the neural circuitry. And it's in a similar manner, for instance, like the cables that we have in our houses that are also insulated and thereby allows then the fast transmission of electricity. We also have these oligodendrocytes and myelin in the brain that then allows information to be transmitted at fast rates. Now, what you have here is a video in zebrafish from a colleague of mine, Tim Schopke, where you can see an oligodendrocyte precursor cell starting to differentiate and produce here myelin around the nerve cells. Now, oligodendrocytes have been thought to have this main function, which is to produce myelin. But actually, when they were first discovered in Madrid in around 100 years ago by Pio del Rio Ortega, actually, morphologically, oligodendrocytes in the brain were seen to have at least 4 types of morphology. And throughout these 100 years since then, also other studies have shown, for instance, that in the different parts of the brain, you can see oligodendrocytes with different kinds of morphologies. However, the main function of oligodendrocytes has been ascribed to be the one producing this myelin insulating layer. Now, we were very interested in my lab to investigate if this morphological heterogeneity could also translate in molecular heterogeneity. And for that, we have applied single-cell transcriptomic technologies. And just to give you a little bit of an overview, so before, many years ago, when we would try to study the brain at the molecular level, we would basically have to dissociate the brain and then start doing the analysis. And I can make a parallel here with a smoothie, that you basically have all the fruits and you put the fruits together, and then you would, the process to analyze the brain, it would imply that you would have to mix it, all the samples together. And that, because of that, when we were doing this analysis of the brain, we could see like that there were like, as in the smoothie, there is a flavor of pineapple, a flavor of orange and of strawberry and so forth, but you could actually not see the properties of these individual fruits. And this is what single-cell RNA sequencing and transcriptomics allows us to actually analyze the brain and other tissues in the body, but to see, instead of having this mix of properties, we can see the properties of the individual components of the brain. So single-cell technology actually is a very recent technology. It arose in 2009, only 11 years ago. And this was spearheaded by different parts of this technology that allows first either to isolate the individual cell in specific microfluidic compartments or in droplets, then also the optimization of the amplification methods of the RNA, and also Illumina sequencing technologies that also allow then to detect, to amplify and detect these RNA molecules of the individual cells. And as you can see in this 11 years from the when in the beginning you could analyze a very few number of cells, now we are in the range of millions of cells. And I would want to also highlight that some of the first studies doing single-cell RNA sequencing were actually pioneered here by the labs of Stan Linnarson and also Richard Sundberg here in Stockholm at Karolinska Institute. So my lab collaborated with Stan's lab in order to investigate the heterogeneity, molecular heterogeneity of oligodendrocytes. And this was work spearheaded by Sweli Marks, at the time a postdoc in my lab, and Amit Saitzel, a postdoc in Stan's lab. And then basically we used the mouse brain, and again use this technique that we could isolate single cells in these microfluidic devices, and then analyze the transcriptome of all these individual oligodendrocytes. And what you can see here is a t-SNE representation of the data. So this data is very multidimensional because you have all the individual genes that are expressed in the individual cells, and one needs to reduce the dimensionality of the data. And this is a way to do it. And here every single dot represents a cell. And cells that have similar transcriptional profiles will be close together in this dot, in this representation. And what you can see here is that, for instance, for the mature oligodendrocytes, we could detect up to 6 distinct transcriptional states, indicating that, which could suggest that oligodendrocytes, the mature oligodendrocytes might have much more functions than the one that I've presented in the beginning, which is only to produce the myelin. And this is something that we are actively investigating in my research group. Now, we are also very interested in the role of oligodendrocytes in multiple sclerosis, because in this disease, in multiple sclerosis, basically what you have is an autoimmune attack where immune cells attack both the myelin and the oligodendrocytes, leading then to this demyelination. And as I've explained in the beginning, what this then causes is that there is a slowing down of the transmission of the electrical impulses in the neural circuitry in the brain and in the spinal cord, which ultimately leads to the symptoms of MS. But we also know that in the brain and the spinal cord, there is progenitor cells, like I showed you in the beginning, can differentiate and produce new myelin. And actually that can happen in the context of MS, and particularly in the relapsing remitting patients, where the patients have the symptoms, but then have a recovery. But however, many patients, they actually fail to have this recovery. And this is partially due to the fact that these progenitors, either they are absent from the lesions, or even if they are there, they are not able to produce new myelin. So multiple sclerosis is a disease that affects around 2.3 million people worldwide, and most of the common symptoms are like fatigue, numbness, blurred vision, weakness, poor coordination, pain, depression, problems in memory, and so forth. They have several different disease courses, as I mentioned before, and they have higher prevalences in higher latitudes and also in Western countries. Now, in the last 20 years, there has been a dramatic and spectacular advance in research and also in treatment of multiple sclerosis, with several treatments that have been available, starting to be available for patients. And most of the treatments actually target the immune system and try to prevent immune cells to attack the myelin, and for instance, try to prevent the immune cells to access from the periphery into the brain. A new class of new therapeutics that it's now in clinical trials is actually to try to stimulate the progenitor cells that I mentioned before to produce new myelin, and then and do regeneration of this capacity to transmit electrical impulses. So this was something that we were very interested in our lab, and we wanted to see how the transcriptome at the single cell level of progenitor cells was affected in the course of multiple sclerosis. So that Anna Falcão, a postdoc in the lab, and David Van Bruggen, a PhD student in the lab, that decided to apply this single cell technology to a mouse model of multiple sclerosis, and they at the peak of the disease, and then focusing on the oligonucleotide populations. And again, this is a representation of the data as you've seen before. And within the oligonucleotide populations, both the progenitors and the mature oligodendrocytes, Anna and David found that there were cells that actually had a specific kind of gene signature of transcription. And particularly, they were expressing genes that are usually only expressed in the immune cells, like major histocompatibility complex class 1 and class 2. And these genes are usually expressing genes that are presenting antigens within the immune system. And it was quite surprising that when we saw that they were also expressed in the oligonucleotide lineage. This is now, if you go to the tissue and look at specific lesions, you can use this kind of technologies called RNA-scope, where you can see the expression of the different genes. And what you can see here in red, you can see this MHC class 2 marker, which is a marker for an immune cell. And in white, you can see the marker for microglia for an immune cell, IF1. And then you have here in green a marker for oligodendrocytes. And then you can see this cell here that it's green, so it's an oligodendrocyte lineage cell. But it lacks IF1, so it's not an immune cell, and it has CD74, so this marker of these MHC genes. And Anna and David looked in these lesions in this mouse model of multiple sclerosis, and they saw that around 4% of the cells that of the oligonucleotide lineage seem to acquire this immune state. So our data using this single cell omics indicates that this progenitor oligonucleotide lineage, apart from the function of producing myelin, can in the context of multiple sclerosis and inflammation acquire this immune oligonucleotide or immune phenotype. And we published this a few years ago, and there are several studies subsequently that have shown again that this co-option of this oligonucleotide lesion changing into immune phenotype. And this is something that now we are investigating very actively in our lab. What does this mean for the etiology of multiple sclerosis, for the maintenance, and possibly also for treatment? Now, Professor Danislo mentioned before epigenetics, and actually this is something that we are very interested in the lab, is to see how oligodendrocyte starts expressing immune genes. And for a gene to be expressed, actually the genome is very compact in the nucleus of the cell in a very close chromatin conformation. But for a gene to be expressed, this protein RNA polymerase has to access the transcriptional start site of the gene to initiate transcription and then activate this transcription. So a few years ago, the laboratories of William Greenleaf and Howard Chang developed a technique called ATAC-seq that uses an enzyme that actually can access these parts of the genome that are open and that RNA polymerase can bind, and then insert adapters in these regions, and then we do Illumina sequencing to see which regions of the genome-wide are open. And so Mandy Meyer, PhD student in the lab in Nagasaki, had decided then to apply this technology at the single cell level, again, using the same paradigm as before, using a mouse model of multiple sclerosis and focusing on the oligonucleotide lineage and using different modalities. Again, this is a representation of the data with, like I've shown you before, but here what you can also see in this genome browser view, this is an immune gene, and this is the promoter region of this immune gene. And what we could observe was that actually the chromatin was already open in many of these immune genes, indicating that oligodendrocytes are actually primed or prepared to start expressing these immune genes upon being in the right environment. This also prompts us actually to investigate genes that are associated with susceptibility for MS. So a few years ago, the International Multiple Sclerosis Consortium published a list of genes that of single nucleotide polymorphism, SNPs, that are associated with genes that might be involved with the risk of developing MS. And many of these genes are in the neighborhood, are expressed in immune cells. And this is one of the reasons that we also think that multiple sclerosis is mainly a neuroimmune disease, autoimmune disease. But what we found with the single cell omics approach is that actually many of these open regions of the chromatin in oligodendrocytes or astrocytes and so forth are actually present in genes that are overlapping with these SNPs, which could then suggest that the susceptibility for MS might not only be revealed in these immune cells, but also in cells in the brain as oligodendrocytes. So we are continuing to explore this, and most of the data that I've shown you was in mouse, but now, for instance, a few years ago, we were starting to do the first studies of single-cell transcriptomics in post-mortem material from MS patients, and together with Anna Wilson, Charles French Constant University in Edinburgh, and Diraj Malhotra in Roche in Basel. But now we are actually extending to much larger cohorts of patients so that we can also correlate with our epigenetic data and try to find if there is novel mechanisms for the disease. So to summarize my talk, using this new technology, single cell omics, both at the transcriptome and epigenomic level, we've been able to uncover new aspects of oligonucleotide biology, both during development, but also in multiple sclerosis. And here we can observe that we have observed this immune phenotype that can acquire, and we are investigating very actively in the lab what does this mean for the etiology of MS. And also, which I didn't have time to present here, we actually also started investigating this at the level of histone modifications that also occur at the epigenomic level. And with this, I would like then to finalize my talk by thanking my group that has been working in many of these projects that I've presented here, and also my collaborators, and in particular the funding agencies. And I would like particularly to mention the Ming Wai Lau Center of Reparative Medicine, which has a Hong Kong and the Stockholm node, and I'm in the Stockholm node, and also that has been funding our research. And with that, I would like to finalize, and I would be very happy to answer any question that you might have.
Professor, thank you so much. Wow. It's really mind-blowing, isn't it? More than 2 million MS patients in this world. All right, now is the Q&A session. Now, because of time limitation, we will again take 2 questions, one from online and one from the floor. Okay, let's take the first question from online. Okay, Professor, I will read this question very slowly. Is there an eureka moment in your work where suddenly everything makes sense? Would it be when your team found that the white blood cells attacked oligodendrocytes in MS sufferers?
I think that characterizes a little bit how research is. I mean, we have many moments where we think it's a eureka moment, and that's like we find something in our research that it's like a completely different from what had been previously described, and that we think, okay, this really can open a new view on the field. And I think the eureka moment for us, it was actually when we start seeing the expression of these immune genes in the oligonucleotide lineage. And as all eureka moments, they are also followed by like doubts. Is this really what we are seeing? And is this a technical artifact? Is this for real? So then it is followed by a lot of technical validations and further explore. And then when we confirm this is actually happening, this is very gratifying. And then we can also, it is also very gratifying when we see others, like other publications from other groups, also seeing that the same thing that we have seen for the first time.
Excellent. All right, next question. We're gonna take it from the floor here in Hong Kong. Oh, yes, my friend there in the middle row. You may have to talk a little bit slower, so for, okay.
So thank you, Professor, for your presentation. My question is, how does your knowledge in biophysics assist you in your research? Because in your study presented, most of them are like molecular biology. So where does physics comes in?
I would say, so I am a biochemist from training, not so much of a biophysics, but more biochemistry. And this has actually been very useful in our research, because our research goes all the way from the basic science to the translational side. And all the mechanisms that we are identifying are biochemical mechanisms by which the position of these histone marks at the genome that regulate whether a certain gene will be activated or not. So I would say that not so much biophysics, but actually biochemistry has been very, very useful in our discoveries.
Are you thinking of changing a major now? No. Okay. All right. Thank you. All right. That was wonderful. Thank you very much. Professor, please don't go away. Stay with us, because very soon we will invite you back for the panel discussion. All right. Okay. So, as I said, panel discussion time. Now, before we invite our guests onto the stage for the panel discussion, just a quick reminder that after the panel discussion, there will be Q&A as well. All right, today we are in for a treat, because for our moderator, we have Professor Cui Lapti. He's a renowned molecular biologist. He was a pioneer in the field of genomics. Professor Cui won international acclaim for his work on identifying the defective gene that causes cystic fibrosis. He had made significant contributions in the mapping of chromosome 7. Professor Cui's research paved the way for Human Genome Project and the HapMap project as well. His work spanned across education, and he was the 14th Vice Chancellor of the University of Hong Kong. He's currently the President of the Hong Kong Academy of Sciences and a board member of the HKLF. So with such heavyweight as a moderator, we're definitely in for a treat. So ladies and gentlemen, please welcome Professor Cui to the stage as a moderator. And of course, we would like to invite Professor Low and Professor Hung as well to the stage for the panel discussion. And of course, we have Professor Castello Branco joining us online from Sweden as well. All right, gentlemen, please take a seat. Okay. We're very lucky that we have all these great scientists in front of us, helping us, fighting for a better future. All right, Professor Cui, the time is yours.
Thank you very much. Now you know that this has not been rehearsed. So I don't know. Yeah, I like to see the third speaker who's on that side. Okay, all right. That's why I was asking where should I sit. Okay, anyway. So there were a number of questions on this huge iPad. It's already prepared. I didn't know that I didn't get to ask questions myself, but since I'm the chair, so I should ask questions myself. Right? First, to start with, we have three very different speakers, all on different subjects. And so it's very difficult to ask a question that only one speaker can answer. So I have to ask questions that all speakers can answer. But I do have a question, a scientific one, that Dennis, you can chip in as well. Just from the presentation we just heard. Okay, we have Ivan here talking about COVID-19. We have Gonzalez from Sweden talking about multiple sclerosis. And from what I read in the recent reports, actually, COVID-19 patients could develop autoantibodies. This is so-called long COVID-19. They develop COVID-19 autoantibodies against the myelin, causing multiple sclerosis. So can you supplement a bit on that? Either of you? Do you know what I'm talking about? Can you tell the audience a bit more?
Perhaps I can start. In fact, for transverse myelitis or multiple sclerosis-like syndrome, many of these respiratory viruses, including, of course, influenza, and of course now COVID-19, they are able to stimulate and cause the generation of autoantibodies. And these autoantibodies can be targeting at the myelin, and as a result causing like transverse myelitis-like syndrome, or at the same time they can cause other kind of autoantibodies, including, of course, the COVID-19, as I've illustrated, is very good at generating antibody against the interferon. So, and other autoantibody could be generated as well, including, of course, some of the rheumatological diseases. So we have yet to tease out which patients or which subjects are, at risk of developing this kind of antibodies.
Okay.
Well known, of course, is female is well known to develop this kind of antibody. And how it's related to long COVID, we do not know. But one thing we know, of course, is that by early treatment, as I've said, you suppress the viral load and you suppress the cytokine and the antibody generation. That will probably lead to a fewer cases of COVID-19, especially related to autoantibodies.
Well, as you can tell, that I'm a little bit interested in this field. I'm in the same field.
Yes.
Anyway, let me hear from Gonzalo.
Yeah, I can perhaps add, I think one of the interesting things that also I've seen in the literature is that actually this kind of coronavirus can infect neural cells, and in particular oligodendrocytes. I mean, there has been a lot of research during these past few years, and also there's a lot of controversy also to know, for instance, if the virus can pass the blood-brain barrier and then infect the brain cells. But actually, some studies suggest that this kind of virus can infect brain cells and specifically oligodendrocytes. And in these studies that I've seen with coronavirus, and not specifically this one, actually the oligodendrocytes actually start expressing some of these immune genes that we have observed in our studies. So I think, and this is actually an area that it's a big black box, and I think there is a lot of interesting research to be done in this area, is like how, for instance, infection of virus in the brain and neural cells can actually modulate the properties of the cells and make them behave differently. But I think that there is a lot of research in this area to be done.
Perhaps I could also supplement, because we are also interested in looking at the anosmia, the loss of smell and taste in our patients, especially many of the young patients. And we find that from our study, from animal and from human, that we are able to find that basically COVID-19 can invade the neurological cells, especially the brain cells, causing the damage to loss the taste and also smell. And one of our study, of course, is by giving the high dose vitamin A, which we have already published, are able to improve the outcome of this patient with the recovery of the brain cells, and it's proven on the MRI.
Okay, I'm really sorry that I get into science, and then of course, scientists may not agree with each other. So I don't want to challenge Ivan at this time, but to move on to something else. So let me ask a general question. In fact, I was given the iPad with questions on it. I was playing with it, and the question disappeared. So fortunately, I have my own questions. So why did you choose research, especially biomedical research? Is that from the very early days of your lives? And perhaps I'll let Dennis just start first.
I think I do research because it's interesting, because in a way it is unpredictable. I mean, it is a privilege to be able to have the chance to see something that nobody else have seen before.
When did you start?
So actually, my first research project is in my third year in Cambridge.
Oh, so already into university studies. Okay, Ivan, how about yourself?
I'm a late starter. I actually started in 2003 in SARS.
So you guys really want to be a medical doctor, then develop interest in research. Okay, Gonzalo, how about yourself?
I started, I mean, I went to university in biochemistry, so I was interested in research very early on. But I have to say it was also a family. I have one of my older brothers, he's a neuroscientist and a neurologist, and actually that's what brought me into being interested in neuroscience, and then developed a career more combining the biochemistry that I was very interested in, but also the neuroscience that I got to get interested in also.
Right, because we're speaking to a group of youngsters who are not differentiated, meaning that they have not read already a researcher in the lab. Some do, but I just want to know that whether or not your instinct in research came from early days, okay? So apparently you can fall in love with research at a much later stage. Okay, then I'll ask you another question. Maybe start with Ivan, you first. Any unforgettable moments in your research days? Not rehearsed.
Well, I think, as I've said, I think the most, my most vivid moment is actually, I think, in the Amoy Garden outbreak, which subsequently I was first called upon and went to a construction site with Professor Kiwa Yun to see where it could be originate from that. And then subsequently, of course, I went to United Christian Hospital with my two other friends and colleagues and saw the 100 patients within 3 days. And I think up to now is still the moment that possibly have changed my life, because it's that study that we described in terms of the clinical presentation and also with regards to the viral changes that relate to my subsequent research. So that is probably the turning point of my life.
Okay, thank you. Dennis, you want to?
So one unforgettable moment is that after we've done the Down syndrome test, I was thinking, how can I push this technology to a limit? And one limit is, can I sequence the whole genome of the baby this way? And actually, I was thinking about it for about 3 years, but I couldn't think how to do it. Until actually one day I got the idea when I was watching a Harry Potter movie. So that was a very unforgettable moment.
Watching the movie or thought about an idea?
Because that movie was in 3D, in the initial bit. And actually when I was looking at the Harry Potter sign flying towards me, the H actually looks like a pair of chromosomes to me. So that was amazing.
Too abstract for all of us, right? So Gonzalo.
I mean, I think for me, it's, I mean, our science is driven by curiosity, and like whenever we find something that it's unexpected, that's what I think it really becomes a unforgettable moment. So there have been several moments, and this is what drives the scientist. But I think perhaps one of them would be, like I mentioned before, when me and my postdoc Anna at the time, we were looking at the single cell data, and we start seeing this strange transcriptional profile of the oligodendrocytes, and we're trying to see what does this mean. And this is like, I think what drives us all is like seeing the unexpected and see what does this mean and what can the implications of this? And I think we all are living for these kind of moments in science.
Okay, thank you. Since the audience has only 2 questions, I'm going to get questions from here. Or actually, I can continue with my list of questions because these 2 questions are kind of heavy duty. So I leave it a second. So I always say that innovation, ideas are really lots of them out there. So innovation, ideas rarely came from nowhere. There'll be some insights or something you see. So Dennis talking about watching a movie. Okay, he got his insight. Okay, so how about others? Maybe go back to you, Gonzalo. You have any insights from doing something entirely unrelated?
I do. I mean, I bike usually from home here to Karolinska, which is like a 35 minutes ride bike, and that's actually quite a good moment to actually think about outside the box and think about, and that's where actually a lot of the ideas, and unexpected ideas come, because you are completely free and like you can think a little bit more freely about your science. So I think it's definitely important to actually zoom in and zoom out. So sometimes if you are too much zoomed in your research, sometimes you don't see you don't see like the overall picture. And sometimes it's good just to step back and see the forest and see, okay, this is actually where I should focus. And I think it's very important, like seeing movies or biking, to be able to really to decouple from your science and then focus again.
You do that in the winter too? The winter is very long.
In the winter, the winter, I don't bike so much now.
Okay, kidding aside, so you slow down during the winter.
Yes.
In thinking. Sorry. Ivan.
Well, my usual moment is when I'm taking a shower in the morning.
Okay.
It's when very quiet that I can actually very focus. Even a very short period of time, I can think of things.
Interesting, a quiet shower.
Yes, in the morning,
Okay.
as a cold shower.
Okay, and Dennis, you have anything to add besides your Harry Potter encounter?
Actually, in the pre-COVID days, I find that when I travel on a plane to conferences, I mean, in between nap, the time between you sleeping and not sleeping, you seem to enter into a trance, and then suddenly some idea will come up.
Okay.
So I hope to travel again.
So I'm asking these questions, well, not without a purpose, okay? In order to become innovative, or you get ideas, you listen more to other people, or you do something outside of your regular sort of routine. Then you may all of a sudden catch a new idea, okay? By listening to seminars on a entire different subject sometimes, okay? Not necessarily in biomedicine. You should go and hear seminars in humanities and other things, all right? So talking humanities, here's a question from one of the listeners or audience. So question number 1, I read it slowly. Is there an ethical red line where medical science research should not cross? For example, using CRISPR to make designer babies? And how do scientists draw such a line? Who wants to go first? It's an easy one.
Actually, maybe I'll say a bit. I think every field has some sort of red lines. It's almost like, even in daily life, there's so many ways, there's so many things by culture or by law that we could not do. Now, for example, like in my own field, I think one red line is that you've used the technology, NIPT, to do sex selection. So that's why when we invented the method, when the licensing terms, we actually forbid people from doing that. Yeah, thank you.
Okay, so Ivan?
From my view, I think about a few years ago, 2 groups actually published a paper in adapting the avian influenza, the H5N1 and the H7N9, into infecting human. And they're actually able to transmit in the macaque model very easily. And I think that is very dangerous because if for example, a terrorist get hold of this, it can cause a pandemic. So basically, I think this kind of science, even though it is, really cutting edge, but there is a danger to it that it can actually cause problem if it's in the wrong hands.
Okay, Gonzalo?
No, I mean, I think it's very important to always keep a very close dialogue between the scientists and the ethical committees. I mean, science goes very fast and the developments go very fast, and sometimes it's difficult for the ethical committees to keep pace with the developments of science. And then actually it's very important that leading edge scientists are in the ethical committees and that there is a close collaboration and dialogue in order really to foresee what might be the consequences of some of the research that we are doing. So I think it is indeed very, very, very problem to, when as a scientist, to think what can be the implications and then have this close dialogue with the ethical committees.
Wonderful, Gonzalo. Actually, the following question that we follow up with what you just said. This is a question from the audience. How important is it for scientists to step out of the ivory tower of research lab to get your points across and or to gain public trust? So to you, Gonzalo.
I mean, I think that's one of our missions also, like to be able to outreach and explain our science and explain the implications of our science and also how the whole scientific process is. And also like to perhaps it's, one can call it ivory tower, but I mean also to explain that science is not linear. It's a lot of, sometimes we have dead ends and that sort of trial and error, and explain that for also the general public that it's, the whole process is actually not a very straight, I mean, sometimes we explain science as being very easy from A to B, but actually there is a long way to go there. So I think outreach is definitely a very important mission as for scientists in order to be able to explain to the general community what we are doing and what is the relevance. Having said that, it's also important to say that I think science driven by curiosity is very important, and that sometimes we don't know exactly what will be the end point of our science, because science will drive you somewhere else. And like, I mean, in the example of my research, now we are doing more immunology, which we were not aimed to do from the beginning. So it's also quite important to explain this aspect, that it's an unpredictable science, and sometimes we cannot see what it's going to be at the end.
Okay, wonderful. I will let Ivan and Dennis respond very briefly because you're very familiar with the subject. So just one sentence from each of you.
I would say that I work in translational medical research. I mean, it is probably impossible to be in ivory tower, right? Because you have to do something which will benefit the patient.
Great, thank you. Ivan?
I just concur with Dennis. But I think my experience lately, of course, is to overcome the hesitancy in COVID-19 vaccine, which I think education and speaking to the public hopefully will address those issues. So that have to take into account many things, including, of course, the culture and, sometimes religious belief in people that you're talking to. So it's just not pure science.
Okay, so I do have two more questions here, but then the question number 3 is disappeared from the screen. Now it's back. We get inspirations from you, and you are the role models for young scientists. What are your role models, and where do you get your inspirations from? Want to go first?
So I think that I was very fortunate to have many good teachers in my life. And one of the senior scientists that I aspire to is Professor Y.W. Kan, Gan Yue Gao. And because he actually worked in a very similar area to I, so he was a pioneer in DNA-based prenatal diagnosis. And I actually remember when I was a junior medical student, I actually read an article written by him, say from a slow boat from China, talk about his trip from Hong Kong to the US to become a scientist. So I learned from the experience, and eventually I was fortunate enough to know him in person. And so, yeah, so he's my mentor.
Very good, very good. Ivan?
Well, I have two mentors because I have two specialists. I'm specialist in ID and also in gastro. But for my ID specialist, I think my mentor will have to be Professor K.Y. Yun, who have been inspiring since I started doing my research, both in clinical medicine and also in translational research. For my GI mentor, it has to be Professor Joseph Leung, who actually previously was working at CUHK, but subsequently when he was spending a year at Queen Mary, I actually learned a lot from him doing endoscopy. So I have two mentors because of my two specialties.
Okay. All right, Gonzalo, you have any?
Yes, I mean, I think I mentioned before that I had in the family, one of my older brothers that was, it's already a scientist, a neuroscientist, that was one of my role models. But I think also like throughout the career, you find a lot of mentors. And I mean, I had mentors both here in Sweden, in Portugal, and also in Cambridge in the UK that have inspired me a lot. But I think one thing it's important, especially for the young scientists, is actually to talk with a lot of people. See, there is not one scientific path. There is several scientific paths, and you make your own scientific paths. So one thing that I have a lot of role models with that have been scientists, my colleagues here at Karolinska, for instance, that I talk and then I get some like inspiration to do my own path. But one thing that you have to think is also you are creating your unique path with inspiration of all the role models that you see. You have to be unique. That's you don't need to follow always the advice. You need also to think by yourself and then think, okay, this is how I should
advice.
proceed.
The next one, I really like this question. This is really for two local speakers. Would you find it concerning that the career of a researcher is much less stable and risky than that of a doctor? What encouragement can you give to teenagers who are passionate in science to pursue a career in research instead of studying medicine instead? Would you give up your medical practice?
Well, I'll say that how much price are you going to pay for a career which is fun, right? And to me, I'll pay anything for that.
Okay, thank you.
I would not give up my career as a practitioner. The reason for that is because my research is all related to my patients. So if I give up my clinical medicine, I would not be able to pursue my research as well. So it came hand in hand.
Okay, thank you. Gonzalo, you would ask your children to go into medicine?
No, I'm not a medical doctor. I'm a biochemist.
That's
from
why
the
I
beginning.
asked.
And actually, it was a very clear choice in the beginning to go to biochemistry because I was really interested in more in basic research. But I definitely think that both paths are. I mean, sometimes I do miss not having that medical expertise, but then I have collaborators that have that medical expertise and then can complement. So I don't think it's two different paths that are very complementary. You will, as an MD and then as a researcher, get, I actually admire a lot when you can combine both and you can get the best of the two worlds, but you also can also do just a biochemistry or molecular biology career and then collaborate with the right persons that are actually doing research in those other areas and combine. Collaboration is key, I think, in that case.
I cannot agree with you more. I myself is also a basic scientist. I'm a molecular biologist, or you can call me molecular geneticist, working in a hospital. So I'm surrounded by doctors. So I had a lot of collaborators when I worked in Toronto. And I was actually asked whether or not there's any questions from the floor. Any? I see hands, but then I cannot really, I don't know. There are two of them at least. Yeah, there are others? Please hold your hand steady so that yeah.
Hello, thank you for your presentations. They were really interesting. So I think that you were talking about like some illnesses like multiple sclerosis or like cancer detection, something like that. But a lot of the research is on like very small, like molecular biology scale. And sometimes like the interactions in the body may be also like more complex than that. Then do you ever evaluate like interactions outside of that very small mechanism. Is that considered with like other research papers? Yeah, thank you.
Okay, I think to turn it the other way is that the environmental factors working together with your whatever change in genes, right? Is that all right?
I don't know whether it is what you're asking, but my interpretation of your question is basically, I think that different types of scientists, some people, if they like really micro thing, they could work on a crystal structure of a protein. But on the other hand, there's also a spectrum of scientists who like to work on big things, like the epidemiology. You look at the whole population. So I think that, the beauty is that there's a spectrum, and you can always choose whatever that you like the most. Thank you.
Okay, I saw a hand out there and the one in the middle, but I cannot see them now. Yes?
Thank you for
Can you
your
raise your
discussion.
hand so that we can see you?
Thank you for your discussion. And I have a question for all three of the speakers.
Please.
Actually, all four. And I
Me,
want
no.
to know what is the defining feature of a scientist that you think one should have in order to be successful in your respective fields? Thank you.
That was supposed to be my question. So you asked my question, so I asked the three speakers. Okay, what are the qualities? Okay.
Well, I think it's never give up, basically.
Never give
You will
up.
fail a lot of failures before you come to
That's called
eventually
stubborn.
success, yeah. Get stubborn, yes, indeed.
Stubborn good enough?
Well, stubborn, and sometimes you need to take advice as well.
Okay, Dennis?
I think one has to be honest because, if you cannot document the observation clearly and honestly, then you just mislead yourself and mislead the world. And then you need to also have a broad base so you can synthesize pre-existing knowledge and then think together. And of course, you have to be humble as well.
Yeah. Okay, Gonzalo?
I agree with all these qualities. I also think that, one thing is science can be a roller coaster with a lot of ups and downs, and that many times you don't really know what your results mean, but then certainly light turns, you get an idea and then you get a lot of good results, but then it goes down. So you have to be resilient. But I would think that as a scientist, what you really need to have is curiosity. At least, curiosity is really what drives you. And also, like related to the previous question, it might drive you for somewhere that you are actually not expected at all that you would be. In my case, I was never expecting that I would be doing more like immunology kind of research like a few years ago, but that's where science drives you. So you just have to let science drive you through where it goes.
Right. So for myself, I cannot agree with the three speakers more, because I started with studying bacterial viruses and then moved into human genetic diseases. And at the end, I was studying population genetics at the end. Anyway, so, and then the chromosome structure, what they call genome, and so on. But then I think that you pursue science, you are getting into so-called uncharted areas. So no one was there before. So you are studying that. And this is a really pleasure. I think that's what Dennis was talking about, right? No one can take it away from you. But sometimes we do come up with difficulties, right? So stubborn is a bad word, but then persistence is a better word. But then, of course, like I said earlier, you look around for help. You present your results to other people. I like to go to seminars and go to actually conferences to meet people, to talk to people. And then once you get the conversation going, people would volunteer ideas to you. So from an entire different angle that you never thought about. So I don't want to capture the conversation, but then I think there's another hand out there. Was that? No? Yes, please. So we have 53 seconds.
So I have a question for Professor Hung. So according to current studies, I have seen some articles about several COVID vaccines, like both mRNA and inactive activated vaccines, I think that's what you call them, has been shown to like lose strength over time. So theoretically speaking, is there a way to like develop vaccines that like the immune percentage will drop less than the current ones?
Yes, a very good question, in fact. Because COVID-19 is a novel virus, so basically no one, apart from those who are infected, have antibody or robust antibody. And as a result, we could only mount immunity via vaccination. And of course, certain vaccine platform may be slightly better than the other in terms of sustaining the antibody level, or initially have a higher antibody level, or perhaps stimulating more different kind of immunity. But the point is that with the current problem with the Delta variants or variants emergence, then we'll probably be chasing after these variants. So probably the final solution is we have to develop a vaccine that will probably work universally for different kind of coronaviruses. And one thing that we worry about is that the MERS and the COVID will come together and become a lethal and also contagious virus. The other thing, of course, is how to block the transmission. So as I've said, we probably need a combination of vaccine, injectable vaccine together with an intranasal vaccine in order to block the transmission or carriage of the virus. So we have still a long way and basically a lot to learn from this virus.
Okay, so the time is up. What remains for me to do is really thank the three speakers, the three panelists. You really have given us a lot of food for thought.
Thank you very much. Wow, what a great panel discussion, and made it so much more interesting by our moderator, Professor Cui. Certainly it is a very fulfilling day for a lot of us. Professor Cui, would you like to say a few words to conclude the seminar?
Really not much. The purpose of the forum actually is to inspire youngsters who are interested in science in different areas to pursue science as a career. So I hope the speakers had inspired you. And then actually, I don't want to draw anything, an analogy, but it's almost like triggering antibody reactions, okay? But Professor Hong did not go into details about immunology because it's too complicated in the public, and then it'll take years to explain. And besides, scientists may not have all the answers. But to me, it's very simple. You have already this antibody stem cells in your system that is research. You are interested in research. By giving you an antigen, expose you to a new area of studies, all of a sudden you click, and then the light bulbs light up, and you become a scientist. And then scientists, life is very difficult, and like one of the audience asked the question, but you have to be persistent, okay? Your curiosity play a big role in finding the answers. Because like Dennis said, once you're into an area of exploration, then no one was there before. Okay, so this is be so fun to go there and find out yourself. Getting there as the first is great, but helping humankind is really what you want to do at the end. So with that, thank you very much. Thank you to the speakers. Thank you for all listening.
There you go, ladies and gentlemen, one more time for Professor Cui, Professor Hung, Professor Low, and Professor Castello Branco.